All right, Assalamu alaikum. So in the last section of this course, we're going to look at applications of quantum mechanics to biology, to the living system. So we're trying to discuss in these last 4 lectures a field that has emerged in the past 20 years. And that field is the field of quantum biology. So it's an upcoming field, and it hasn't been there for very long, just 15, 20 years. And in order to understand quantum biology, we would like to make a foray today into learning the essentials of quantum mechanics, quantum physics. So today's lecture will be devoted to the essentials of quantum physics. And the kind of phenomenon that have been explained by quantum biology, they are very diverse. For example, processes as important as photosynthesis, how chlorophyll harvests light and converts it into useful chemical energy. A process as important as photosynthesis is describable by quantum mechanics. And the promise that I started off with in this course was I would like to describe how different senses work. So I will focus my attention on the vision in migratory birds. How birds find their path on the terrestrial landscape. How every year they move from the same location in Siberia, come to the same location to warmer lakes inside the subcontinent or in Africa and go back. How do they find the path? The truth of the matter is that the answer lies in quantum mechanics. So we're going to look at vision in migratory birds, and we're also going to delve for some amount of time into the sense of smell. All right, and we'll see how quantum mechanics is able to describe smell. So we look at two kinds of senses. One is a visual sensation. The other is a smell sensation. Okay. An olfactory sensation, yes. Where? Right. So how do they know when to move? So it depends upon, so this depends upon the seasons. It depends upon the temperature. It depends upon the length of the day, the diurnal variations. That is the length of the day versus the length of the time makes the bird understand that now it's the time to migrate. Okay. There are other phenomenon in the human body or in animal bodies that also can be ascribed to quantum mechanics. One of them, for example, is DNA and heredity. The other is the function of enzymes. And one last frontier where quantum mechanics might come to the rescue is consciousness itself. All right, so I put some of these phenomena in yellow and others in white. The yellow ones we're going to touch in this course. And these are relatively well understood. The connection between quantum mechanics And these phenomenon is relatively well understood, although still it's not as well understood as many other phenomena in life sciences or physical sciences, but still we do have a solid grasp on how these phenomenon invoke quantum mechanics. These processes, the ones that I've written in white, they are as yet slightly uh, evocative, and they're also not very well established. So they are at the conjectural stage, I would say, at the moment. So we're not going to look into these. Rather, we're going to spend a lot of time on vision, smell, and some time on photosynthesis. Now, when I was looking at when I was designing the course outline for this course, I had recommended that students who take this course must have taken modern physics, or if they're freshmen, they would be co-registered in a modern physics class, because that's our first exposure to quantum mechanics. So with that idea in mind, I would like to devote today's lecture on just giving the very basics of quantum mechanics from a slightly semi-mathematical perspective. All right. So before I proceed, I would like to ask if you could raise your hand and tell me if you've had some introduction to quantum mechanics before. Everyone, everyone up. Thank you. आप कौन से class में यहाँ पे? So yes, so you've taken modern physics, and what do you remember of quantum mechanics?
Photon, Hamiltonian, potential and solving for a system in Hamiltonian.
Right. So in layman terms, what is quantum mechanics? Just forget about the maths, forget about the Hamiltonians, forget about equations. What does quantum mechanics tell us? What is it about? The first question I would like to answer, like you to think about, is what is quantum mechanics about? So this is the key question. And for then, I would like to invoke why has a biological description evaded quantum mechanics? Why haven't they been come into wedlock? Why has it taken so long for quantum mechanics to put its claws on biology and bring it under its purview? What is quantum mechanics really about? What kind of systems does it deal with? आप बता सकते हैं? Right. So what is the quantum level? That's like a tautological answer. What is the quantum level? Subatomic particles. Something that is really small generally comes under the purview of quantum mechanics. And by small, the scale of smallness is something at the level of atoms, molecules, what's inside atoms, what's inside molecules, how do atoms come together and make molecules. That is generally considered to be the realm of quantum mechanics. Biology, on the other hand, or the human body, on the other hand, looks at macroscopic objects, bigger sized objects. So quantum mechanics generally talks about things that are small, small on an atomic scale, perhaps, or a molecular scale. But when we talk about biological systems or the human body, we're dealing with a macroscopic object. We're dealing with macroscopic cells, tissues, organs, and all those microscopic phenomenon which are describable by quantum mechanics, they are just averaged out. So when you looking at a newspaper print, for example, if the newspaper print is close to your eyes, you can see the individual lettering distinct from one another. But when the piece of paper moves away, everything appears like a smudge of ink. So when you're looking at things from a distance, the average behavior is observed. So when individual particles, although they are described by quantum mechanics, when you're looking at an ensemble, which is a large collection of these quantum particles, you will only see the average quantum behavior. And the average quantum behavior is the classical world. So if I live in a quantum world, and I take many systems that are behaving in a quantum mechanical fashion, and each system is, for example, describable by a wave function, and you all know what a wave function is. And this wave function can be a function of time. It changes with time, for example. Then if I were to look at the average behavior of this ensemble, this collection of microscopic particles, quantum particles, the overall effect will appear classical. So it appears to us that macroscopic finite human beings live in a classical world. But once you get deeper into the level of enzymes, molecules, atoms, electrons, electron pairs, free radicals inside the retina of an eye, it is there where the glory of quantum mechanics starts to unfold, where quantum mechanical principles are required to explain what is happening. So we do not see quantum mechanics per se. And that's why biology has evaded quantum mechanics for so long, because generally we are dealing with macroscopic objects, which have been shaped by the forces of evolutionary biology. Evolutionary biology does not require our brain to think quantum mechanically. We only see average macroscopic pictures. And that's why quantum mechanics is difficult to perceive. All right. So quantum mechanics generally deals with objects that are small and objects that are fast. And another thing that quantum mechanics generally deals with is objects that are really, really cold. Generally, cold for the following reason. A quantum object shows its quantum properties when it is left on its own, when it is isolated. When it interacts with the environment, the quantum properties wither away because the environment is disturbing the system. And the disturbance is stronger if the temperature is higher. So that's why if you were to build a quantum computer, which would be the next industrial revolution, the next big revolution, if you were to build a quantum computer, generally they would like to build it at very low temperatures. The working qubits in a quantum computer are at roughly millikelvins or just a few kelvins of temperature. So quantum mechanics works really well when the system is cold, ultra cold. But our body is at 37 degrees centigrade. It's very hot from the quantum mechanical perspective. So that's why quantum mechanics is difficult to perceive, because the systems that we deal with, they are large in size and they are hot, they are warm. Biology is warm. Nevertheless, there are phenomena that require a fundamental quantum mechanical description. So now we've studied, or we've had a quick survey of what is quantum mechanics about and what kinds of systems does it deal with. We'd now like to look at the basic tenets of quantum mechanics. Any questions in the meantime? The basic ideas. It's probably the first time that I'm going to cover all the basic ideas in just 30 minutes. All right, the first basic idea of quantum mechanics is that any particle or any object, now these objects are quantum objects, these are special objects like protons, photons, electrons, molecules, atoms. Any object is described in most cases by a quantum state, and that is denoted by some symbol psi. Okay, and generally you'd like to put this in a strange looking symbol which is called a ket. Okay, so just like if you have a beaker of water, it has a certain temperature. the temperature, pressure, and volume of that fluid describes the state of the system. Likewise, you have an object which is described by a quantum state which is denoted by psi. So every particle has a quantum state. Now, this psi, when it's inside a ket, is a state. Psi on its own is also a state. There are just two different ways of writing this quantum state. This state could be expressed in terms of position. For example, let me draw a wave. This wave may correspond to an electron. A classical picture of an electron is like a billiard ball. That's a classical cartoonic iconic representation of an electron. Suppose an electron is moving from left to right at a certain speed v. This is the classical Newtonian picture. Quantum mechanics, however, would say, suppose this is in vacuum. It's not seeing any forces. There's no electric field, no magnetic field. Quantum mechanically, this picture is incomplete. Quantum mechanically, you would like to describe this particle by a function, psi. This function is called a wave function. All right. So this function, this axis is now x. The function is with respect to function of something. It's a function of x. So suppose this variable is x. Now this function will have a certain form, a mathematical form. Now what's the mathematical form for a sinusoid? Some amplitude a, some sine, or so let me just put it like this, eikx. Okay, a complex number. Okay, you take the real part of this, you'll get the wave function, or the imaginary part. So this is a function. Since we're talking about an electron or a quantum particle, it's a quantum wave function. Okay, now suppose that you have the electron and you look at this wave, which describes the particle, which describes the properties of this particle, and you take a snapshot in time. You take an image of this wave function in time, at some time. And then you retake an image at some later time. Suppose this wave function has advanced forward at some later time. So yellow represents the wave function at some later time. So this wave function not only has position dependence, it also has time dependence in it. So this is a function of both position and time. Right? So I need to put in some time dependence in here as well. So one common technique is to put the time dependence as omega t. So with x, there is a variable k. What is k related to? If you look at the picture, what is k? It's the inverse of the wavelength. So you look at the wavelength of this wave, that's going to be 2 pi over k, which is lambda. So this k is the inverse of the wavelength, the period in space. Similarly, k is the period in space, and this omega is the period in time. So if you make your position fixed, and suppose when this wave comes in, a measurement device hops. When the wave progresses, the measurement device hops. So this hopping will be at a frequency omega. A wave is coming after every 2 pi over omega. So the inverse of this 2 pi over omega is the time period of this wave. So now we have captured both the position dependence and the time dependence in this wave function. Now this wave function depends upon position and it depends upon time. Now there was an engineer, in fact, Dirac, who said, okay, we have this wave function. Let's put that symbol of the wave function and put it inside the symbol ket. So the ket is the state of the electron, and you can represent this in position and time, and then it becomes a wave function. Okay, so this is a more abstract quantity. This lives in, for the technically minded people, this lives in a Hilbert space. So this quantum object, which represents some property of some quantum particle, be an electron or a photon or a neutron or a molecule or a bacterium, is represented by psi put inside ket. It represents the quantum state of the particle. And then you can represent this, the word represent. You put it in a basis, you represent it in position and time, and it becomes a wave function. So both of these terms can be used interchangeably, wave function and the quantum state. All right, so now this is one idea. Now, if a particle has this wave function, then if I ask you the question, where is the particle located? So a very naive question, which makes all the more sense in our daily lives. Where is the particle? Where is the electron? You can't get a simpler question in physics. Where is the electron? If an electron has this wave function, where is it? What will be your answer? Exactly. The electron, before you make a measurement, before you actually have a device that were able to find the position of an electron, say an electron microscope could be one device, or before you had some measurement, the electron is everywhere, right? But when you make a measurement, you are projecting this, you find the electron located somewhere, say at some x naught. You locate the electron to be somewhere. Okay, you could have found in one measurement the electron could be here, 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 anywhere. But suppose you do a measurement and posteriori, a posteriori, you find out the electron to be located at some position x naught. Then what you've done is, you can't say that the electron was always at x naught. What you've done is you've taken this wave function and collapsed this wave function. Collapsed this wave function at one point, x naught. So before you have made a measurement, this is your wave function. And after you've made a measurement, suppose you get x naught as your answer. After you've made a measurement, the wave function is totally different. The wave function is now the new wave function post measurement. We're talking about post measurement here, after the measurement. This is your x-axis. Post measurement, your wave function is just a blip at x naught. And mathematically, this blip, which is zero everywhere and only exists at this point, is called a Dirac delta function. So now you have created a new wave function. The act of measurement creates a new wave function, right? And there's a certain probability. You always talk about probabilities of whether you would find your electron to be located at x naught, x1, x2, x3. So let me recap, and then I'll take questions. So the first thing is, we have a wave function. Psi. We do a measurement. the act of measurement collapses the wave function. And we get a new wave function. Suppose that new wave function is denoted by phi. So the act of measurement creates a new wave function. So this is something really confounding about quantum mechanics, is that if you have an object, the act of measurement disturbs the system and the wave function collapses into some new wave function. So you have created a new state as a result of measurement. So quantum mechanical states are extremely fragile. Any attempt to measure those states disturbs the state, and probably you might get a new state altogether. All right. Now, the other thing is that we can always talk about probabilities. Which means that the probability that starting off with a wave function psi, you do a measurement, your outcome is phi, is going to be quantum mechanically, don't be baffled if you don't recognize this or don't remember this or don't understand this. But let me just write it once for the sake of completion. There is a probability that upon measurement, the initial wave function collapses to phi is given by the inner product of psi and phi modulo square. And I'm going to explain this in another fashion in a minute. Suppose, let me do an experiment. Suppose I have a device, and now I'm talking about photons, light. So I'm talking about light, which is extremely important for vision, by the way. How do we see? And light, we know, is comprised of these quantum particles called photons. Just like electrons, in the electromagnetic realm, we have photons. These are small particles. Each of them has a certain energy. Okay. You can get photons from laser light. You can get photons from the sun, and so on. In photosynthesis, you're getting photons from the sunlight. All right. Now, I put photons into a device. Light is coming into a device, and there's a beam of photons, one after the other, issued from a laser coming out. And they hit this device. This device is a special device called a beam splitter. What the beam splitter does is that if a photon comes in, it transmits the photon, lets it go straight through with a 50% probability, 50% probability of transmission, and it reflects the photon with a 50% probability. So it's a perfectly balanced beam splitter. It splits the incoming beam into two parts, transmits and reflects with equal probability. And we've engineered it such that the probability is always equal. Now what I do is I place some kind of detector here and some detector here, identical. Both of them are identical detectors. Let's call this D1. Let's call this D2. And suppose that these detectors are special detectors. We have them in our lab as well. That when a single photon hits the detector, single photon, it gives an electronic pulse. Just one pulse, say 10 nanosecond, 20 nanosecond pulse. So you count the number of pulses, you can count the number of photons. So it gives an electronic pulse when a photon hits on it. And it's a single photon detector. It detects a single photon. It has that capability. By the way, the human eye The retina of the human eye can detect 40 to 50 photons. That's its threshold of detection. There are certain marine animals that can detect even a single photon. Anyway, so these are detectors. Now, if I have a laser source and this laser is extremely low intensity, and I can achieve that by putting in certain absorbers in its path, and so low an intensity that only one photon comes out at a time. So one photon comes in, passes through the beam splitter, and then you have to wait for the next photon to reach the beam splitter. So it's a highly attenuated beam. Highly attenuated beam. So at one time, there is only one photon inside the beam splitter. Okay, so this kind of experiment is called a single photon experiment. One photon at a time. We're doing a single photon experiment. All right, so single photon comes in. The photon cannot be split. It's a fundamental particle, it cannot be split. In a linear process, the photon just remains a photon. Okay, like an electron cannot be split. Now, if this were the case, what's the probability that D1 clicks? 50%, and this probability. So the probability that D1 clicks is 0.5, which is equal to the probability that D2 clicks. And both of these photons, both of these detectors cannot click at the same time. There can be no simultaneous detection or no coincident detection on the two detectors. One precludes the other. One detector precludes the other or excludes the other. So because the photon cannot be split. All right. Now let's, this is, suppose the incoming state is psi of a photon. And I call this path, I give this path a quantum label. This path is a quantum state. So now I am labeling, or what do you call it? I am encoding the quantum state in the path that the photon takes. Let me rephrase this or repeat this. I'm encoding the quantum state in the path that the photon takes. So this path represents one quantum state of the photon. This path represents another quantum state of the photon. Right. So now in computer science, you are used to bits, zeros, ones. Let's call these q bits, quantum bits. So let's call this state ket 0. Let's call this state ket 1. All right. So now you have some quantum state coming in, and this produces two possible output states, but only one output state is observable at a time. Now another, I would like, now I'm going to take you to the idea of superposition. So now you understand this. Any questions about this? All right. Let's move on. I think this basic understanding will help clear a lot of things about quantum mechanics, and I'll try to live to my claim of covering all the basic ideas in just 40-50 minutes. Now, suppose I have the same system. This is my first beam splitter, B1, 50-50 beam splitter. This is my identical second beam splitter, B2. And these two devices here are perfect mirrors. So any photon that hits on these mirrors is reflected with 100% efficiency, with 100% likelihood. Perfect mirrors. So these mirrors are perfect. Now a photon comes in. A single photon comes in. This is my single photon coming in. By the way, before we move on to this, if we look at this beam splitter, how would I know that the probability is 50%? So I just put in a single photon. It either goes here or here. How would I know that it's 50%? I would have to repeat the experiment, right? I will have to have many single photons come in one after the other, but with certain time delay so that only one single photon is in the system at a time. So in order to build the statistics, I'll have to repeat the experiment on multiple copies of single photons. Just if I give you a coin and you're not allowed to see the coin, both sides of the coin, but you're allowed to throw it and see whether you get a heads or a tail. You can't tell in one go whether the coin is balanced or fair. You'll have to do the experiment multiple number of times with multiple identical copies of the coin to find out what the probabilities are. So in order to build the statistics, you'll have to do multiple runs of the version. By the way, there's another theorem in quantum mechanics which says that you cannot clone quantum states, but that's beyond discussion here. Anyway, if you were to repeat this experiment with this setup, what's the probability that D1 clicks? And what's the probability that D2 clicks? Both of them are identical beam splitters. Should be 0.5, 0.5. This is our classical prediction. All right. Now, you can imagine each beam splitter to be a coin toss, right? Because it produces heads and tails with 50% probability. But when you perform this experiment in the laboratory with real photons, you observe that only one of the detector clicks. So this is a baffling observation. Right? And it's true, it happens in the laboratory. You can walk down to the first floor. I have a laboratory that demonstrates this. Only one of the detector is going to click. Suppose what you really observe is that the probability that the detector D1 clicks is 100% and the probability that the detector D2 clicks is 0 within the experimental uncertainties. Now, how do you explain this? G, G. Exactly the same detector. हमने वहां से उठा के उन detectors को उठा के इधर रख दिया. Exactly the same detectors. All right. Now, quantum mechanics describes this. And quantum mechanics describes this by invoking a really important concept that you can't see in classical physics or classical electronics, and that is the idea of superposition. I'm going to explain this. Now, what is the idea of superposition? This is really important for quantum biological systems as well. So let's investigate this idea in the next 5 to 10 minutes. Any questions? नींद आ रही है? मुझे भी आ रही है. जी. जी
कि जो रोशनी की रफ्तार है वो एक जैसी तभी भी
ये रोशनी की रफ्तार तो एक जैसी ही है, मफरूजा नहीं है, ये हकीकत है।
we cannot go
नहीं लाइट नहीं, इट्स नॉट अ थियरी। लाइट हैज दिस फिक्स्ड एब्सोल्यूट स्पीड, यू कैन मेजर इट, people have measured it all over the ages, right? So now how do we describe this? So we describe this by first of all having this idea of a superposition. And so I look at a beam splitter. So if a photon comes in into the beam splitter, incoming photon, the incoming photon is in some state ket 0. And these are two output ports. One of them is ket 0, one of them is ket 1. What the beam splitter does is that it takes an input. Do you know what a logic gate is? So classically a logic gate, what is can you give me an example of a logic gate? And gate, right? Anything that, so the problem with the AND gate is that it has two inputs and one output. Can you give an example that has the same number of outputs as inputs? Inverter. So it inverts. So what the inverter does, basically, I think this is a symbol, takes a 0 and outputs a 1, takes a 1, outputs a 0. If this were a quantum gate, I could write a truth table. So I could put an input and output. If the input were ket 0, the output were ket 1. If the input were ket 1, the output were ket 0, right? So you can write a truth table if this were a quantum gate and the states, input and the output were quantum states. Now the beam splitter also acts like a gate, but this is now a quantum gate. And what this gate does is that it has a certain truth table. Okay, so let me write down the truth table for the beam splitter. So here is the truth table for the beam splitter. Input, output. So if I input ket 0, I get a superposition state, which is I get ket 0 plus ket 1. We need to divide this by under root 2 as well for normalization. I'll explain this later, but just remember that there's a under root 2 as well for normalization because I want to keep the probabilities adding up to 1. All right, so take my word here for the time being, for the next 1 minute. And if the input were ket 1, I would get ket 0 minus ket 1 over under root 2. Now these states here in this column, they represent superposition states. The quantum state is a superposition of ket 0 and ket 1. I don't like to use the word mixture here. Mixture is not a good word because mixture is something very soupy and very random. It's a superposition. It's a superposition of ket 0 and ket 1. Now this photon, now the naive minded should think of this in the following manner. And this is how in popular science books, popular quantum mechanics books, the first they represent this idea is that after this photon has gone through the beam splitter, it takes up both paths at the same time. Both paths. It's in between the two paths everywhere, unless you have made a measurement. All right, it's in both paths at the same time, and you can't tell which path it has taken. Right. So I'm going to now explain this phenomenon in terms of the maths that in terms of the truth table that I've introduced over here. Now, where do I go? I need this. I just have to. No choice but to go here. So let me redraw that particular diagram. All right. Beam splitter 1. A photon comes in here. What this beam splitter does, it produces a superposition. So this path is called cat 0. This path is called cat 1. So my input is cat 0. After the first beam splitter B1, I obtain a superposition cat 0 plus cat 1 over under root 2. Remember, I'm not making any measurement here. I'm letting the photon run its full course. I'm not disturbing the system. I'm not collapsing the wave function. I'm not looking at the superposition. Remember, I'm not looking at the superposition, but there is a superposition. Now, this is kind of a paradoxical statement, but believe in me that this is going to explain what you observe. So here, this photon is everywhere. It's in the two arms of the interferometer. This device is called an interferometer. Interference is an important phenomenon. Quantum interference, important phenomenon in quantum biology. So here, this photon is is in both arms of the interferometer. It's in a superposition state, but you can't tell what arm is it in. So now, when this field, this spread out photon, just like the electron that was spread out in space, this photon field that is spread out in space in both arms of the interferometer, what happens when it sees the second beam splitter? So now this state is being input into the second beam splitter. So this 1 over under root 2 is just a common factor. Now something happens to cat 0. Cat 0, if you look at the truth table, once again goes into a new superposition state, right? So cat 0 transforms into this. This is what happens to this object here. And what happens? So this plus sign comes here. What happens to this cat 1? This cat 1 is transformed by the beam splitter into this state. So now we started off with the superposition. And then we had another element, another beam splitter act on the superposition. And let's see what final state do we get. Now there are two possibilities, get 0, get 1. These are the two output paths. And we have a detector here and a detector here. And we're looking at the probabilities that what's the probability that D1 fires and what's the probability that D2 fires. So we're looking at the state after the beam splitter, just before the detectors. So here, if you observe, this cat 1 just goes away. There's a minus sign here. And you left with cat 0 plus cat 0, 2 under root 2, 1. So all of this, you just do all the cancellations. Your output state is cat 0. And the probability that the detector D1 clicks is 100%. And the probability that the detector D2 clicks is 0. And this can only be described if in the two arms of the interferometer, before you made the detection and before you have let the photons impinge on the second beam splitter, you have a superposition. So a superposition must exist here. And if now you might be really cunning, you might be the devil's advocate, and you would like to say, okay, if a superposition exists, and if it does exist, let's try to measure, let's try to put a detector here, right? And see which path does the photon take. But if you attempt to do that, suppose you put a detector here. Now I'm representing the detector by the human eye, right? So this is some kind of detector, some kind of particle here. So you put some electron here or some small particle here that when photon strikes on it, light is scattered and you can look at it in a microscope. So this is like an eavesdropper, someone who's trying to peep into the system, peep into the black box. कि घर में बच्चों के लिए कोई तोहफा आया है और बच्चे से सब्र नहीं हो रहा, वो बच्चा खोल के देखने की कोशिश कर रहा है कि उस तोहफे के अंदर क्या है. ठीक है, वो देखना चाह रहा है कि श्रोडिंगर कैट जिंदा है कि मुर्दा है. ठीक है, so trying to peep into the system before the actual drama has unfolded. And when you do that with some kind of mechanism, suppose this is an electron, when a photon comes in the vicinity of this electron, the photon is scattered by Compton effect and it's observed in a microscope. So if this microscope clicks, it means that the photon is in this state. So if it sees a photon in this state, it means that the photon has taken this path ket 0. And when it has taken this path ket 0, the photon gets destroyed, right? Because the photon has hit the electron and it is scattered and produced new photons and it's destroyed, right? But when this detector does not click, then the experimenter would say, no, the photon is now in the state ket 1. But when it is in the state ket 1, these probabilities transform back to their classical values, 50% and 50%. So the quantum superposition has collapsed. So the act of measuring the superposition collapses the state. And you're not able to get this quantum interference effect. Remember, notice that this is an interference effect. This is a destructive interference for ket 1 and a constructive interference for ket 0. That's why this process is called quantum interference. So the key idea is that in quantum mechanics, superpositions exist. And if you want to be over clever and over smart and try to find out that a superposition exists and try to come with the lofty claim that you will find a photon in both paths, that's not possible. And if you do find out which path the photon takes, then this quantum interference collapses, it washes away. And you no longer see any quantum effect. You see just the pure classical effect. Now, Feynman describes this with his famous interference experiment. And I think you're all aware of that. So I'm just going to draw a diagram for that. By the way, some really interesting thing about interference. If in this experiment, you put in one of the paths an object, what this object does, some medium, some piece of glass, some medium, this object also has some truth table, right? Suppose this medium, I call it a phase medium. And what this phase medium does is that it has a truth table, and the truth table is of the following fashion. Ket 0 comes in, nothing happens. Ket 1 comes in, the output is EI phi ket 1. So this input incurs a phase phi, right? So the nice thing about quantum mechanics is that you can have complex numbers into your quantum states. Remember, here at the top, you have a complex number as a wave function. So if this were the case, let's see what's going to happen. Ket 0 becomes this. Nothing happens here. You get a superposition, but then this superposition transforms to the following: ket 0 plus ei phi ket 1 over under root 2. A phase factor is accrued with ket 1, right? According to this truth table here, right? Now let's see what happens after the second beam splitter. By the way, this is quantum computing 101 as well. This is a 1-qubit quantum computer, simplest quantum computer you can think of. Now, ये बात समझ आई आपको? देखो ket 0 के साथ कुछ नहीं हुआ जब वो इस यानी इस पूरी सुपरपोजिशन पे ket 0 remains unchanged but ket 1 accrues a phase. तो ket 1 इसके साथ एक phase factor लग जाएगा. Now all of this is still a superposition where the relative phase has changed between ket 0 and ket 1. If this phase becomes pi, this becomes ket 0 minus ket 1. But it's not necessary to have ket 0 minus ket 1 or ket 0 plus ket 1. You can have an arbitrary phase as well. Okay, now when this superposition state passes through the second beam splitter, once again I invoke the truth table of the beam splitter. Ket 0 becomes ket 0 plus ket 1 over under root 2. And this ei phi remains where it is. And this ket 1 becomes ket 0 minus ket 1 over under root 2. Okay, let's now do some algebra and simplify this a little bit. That will be our last undertaking for today. So this becomes 1/2. With ket 0, I have 1 plus ei phi. And with ket 1, I have 1 minus ei phi. All right. Now, another cunning thing that I could do is I could take ei phi by 2 as common. Got it? This is my output state. Now, what's this? This is 2 times cosine phi by 2. And what's this? This is minus 2 iota sine phi by 2. So this 2 just fades away into oblivion, and I'm left with this state. Now, now this is a state that is coming out of the second beam splitter. What the first detector does is that it projects this state onto ket 0. So I have 2 detectors at the output D1. This path is ket 0, this path is ket 1. The probability, now this is a postulate of quantum mechanics. The probability that D1 clicks, if I know what the state is here, let's call that state psi. And this is my psi. It's in a superposition state. And I want to find out the probability that the detector D1 clicks. How would I do that? I would take this state psi and overlap it with this state. And I put it in this notation, which is called a bra. I put it inside the bra, which is another way of saying that I take this quantum state and find the dot product with this state. I find a projection onto this state. And I then take the modulus square. So now I have this state here, psi, and I have certain rules. If I take ket 0 and find its overlap with itself, this is like a vector dot product with itself. What do I get? A vector अपने ऊपर ही dot product लेंगे उसके साथ 1, provided the length of each vector is 1, right? So the dot product of a vector with itself, which is a ket and a bra, is the overlap of a ket and a bra. This is 1, okay? But ket 0 and ket 1 are mutually orthogonal. If a detector D1 clicks, detector D2 cannot click. So they are mutually orthogonal, which means that the dot product between them has to be 0. So if I were to take ket 0 and 1, or this thing, the answer would be, oh, sorry, 0, 0. Now I use this rule. So I have this psi, and I want to calculate this thing over here. So EI, so this thing EI phi by 2 cosine theta by 2 ket 0 minus I sine theta by 2 ket 1. This is my ket here, right? Ket psi. And I find its overlap with bra 0. So I put a big bra 0 here, okay? And at the end, I take the modulus square. So the overlap of ket 0 with ket bra 0 with ket 0 is 1. But the overlap of this bra with this ket is 0. So it vanishes. So I'm only left with this term here. This term, this coefficient here. And what's the modulus square? The modulus square of this term is 1 because it's a complex number. And I get, my answer is cosine square phi by 2. And if I were to repeat the same calculation and find the projection onto this detector, the answer will be sine square phi by 2. So this probability D1 is cosine square phi by 2. And the probability D2 is sine square phi by 2. And these probabilities, by the way, add up to 1. So you can do this as a homework, find out this probability. And that's why we have these 1 over under root 2 factors and also that these probabilities add up to 1. So the idea is that we have an interferometer. If I were to change phi and I were to plot PD1, I will get a cosine square graph, like an interference fringe. And if I were to plot PD2, I will get another interference fringe, which is 90 degrees out of phase. So by controlling phi, I can choose which detector does the photon land onto. And all of this is only possible if I were to assume that in the arms of the interferometer, a superposition exists. Now that's the strange thing about quantum mechanics. Superpositions exist. If you're a computer scientist, you have logic 0, logic 1, bits, zeros, ones, that's it. You cannot have a transistor which is 0 plus 1. But quantum computer, quantum register, if you are dealing with not with bits, but with qubits, you can have the superposition. You have 2 qubits, you can have a superposition of 4 states. 3, you can have superposition of 8 states. N qubits, you can have an exponentially large number of superpositions. And that is the idea of quantum parallelism, which gives quantum computers their power. Now, all of this is directly related to quantum biology. The only last thing that is left over is the idea of tunneling, quantum mechanical tunneling. It is very important for biological systems. But I think with this basic recipe, with this basic introduction that I could give at 100 level in just one sitting, I've tried to refresh your ideas on quantum mechanics, and we'll use some of these concepts in the later classes. All right, thank you very much.